And this is the process of transcription in prokaryotes. The first one is the initiation phase, and the second one is the elongation phase, and the third one is the termination. So, initiation, as we all know, this is the starting phase. Elongation is the continuous phase, and the final one is the termination. So, here only your transcription stops. Okay. When coming to the initiation phase, RNA polymerase recognizes the promoter region and starts the transcription. So, where the promoters which will go and bind to your template DNA, at that region only your transcription will start. Okay, so that is the starting point of transcription. In that process, RNA polymerase should recognize the promoter region. So, I will explain the promoter region in later in this video. And the second one is the elongation process. The RNA strand is continuously growing. So, in this case, the newly synthesized, not newly synthesized, it is going to synthesize. So, the process will continuously it will go on. So, the RNA strand is continuously growing. And the final one is the termination process. The RNA polymerase stops synthesis and the nascent RNA is separated from the DNA template. What is the nascent RNA? Which is nothing but our newly synthesized RNA is called as a nascent RNA. Okay. And initiation of transcription at promoters. Transcription is divided into three steps for both eukaryotics and prokaryotics. The process of elongation is highly conserved between prokaryotes and eukaryotes, but initiation and termination are somewhat different. Okay. So, what they stated means elongation is more or less similar in both prokaryotes and eukaryotes, but in case of initiation and termination is somewhat different in both the eukaryotes and prokaryotes. So, initiation. So, as I mentioned already, for initiation process, the strand has the promoter sequence. Which strand? Your template strand has the promoter sequence. Automatically, the RNA polymerase, which is designed for special nature, is to recognize. The word is very much important, that recognize the promoter sequence. So, and then only the initiation starts. So, RNA polymerase recognizes the TTGACA region -35 sequence and slides to the Tata at region -10 sequence. Then opens the DNA duplex. Okay, so what this stated means, as I told you already, the RNA polymerase, the enzyme which will recognize your promoter sequence. The promoter sequence has the TTGACA nucleotides, okay, at the upstream sequence, minus, which denotes the upstream 35th nucleotide, which is upstream present, upstream 35th nucleotide, which is TTGACA region, and slides to the TATA at TATA at the region of upstream, minus, which denotes the upstream. If in case the plus is present, means which is downstream. Okay, so -10 sequence upstream TATA region. So in that region only it will the RNA polymerase which will go and bind in that region and then opens the DNA duplex. See here. So, if the promoter region is there, the RNA polymerase automatically recognize. Why we need to recognize means this polymerase will check whether these nucleotides are present or not. After that only it will go and bind there and then it will opens the DNA duplex. So, here the duplex will unwind. So unwound the DNA. 17 base pair open. So here 17 base pairs opened. So hope you cleared. Okay. Next, the first nucleotide on RNA transcript is always purine triphosphate. Okay. GTP is more often than ATP. GTP means guanine triphosphate. So this is more often. More often means repeated. Okay. அடிக்கடி இது when compared to ATP, the GTP is quite oftenly present. Okay. And the pyrophosphates structure remains on the RNA transcript until the RNA synthesis is completed. The three molecules from transcription initiation complex. RNA polymerase. So, as I told you already, RNA polymerase is one of the major enzyme which will recognize the promoter sequence and then the DNA duplex will unwound. So, that much only you clear. So, this is the structure of RNA polymerase and this is the subunit. Okay. So here no primer is needed for RNA synthesis. The alpha subunits fall off from the RNA polymerase once the first 3 to 5' phosphodiester bond is formed. Once the phosphodiester bond is formed, the alpha subunit which will automatically fall off from the RNA polymerase. C. The core enzyme moves along the DNA template to enter the elongation process. So, the alpha subunit which will automatically removed from the RNA polymerase when the 3 to 5' phosphodiester bond is formed. So, after what has happened, the core enzyme which will move along the DNA template to enter the elongation process. Okay. So, before enter into the elongation process, I'll show you the DNA polymerase structure so that you might know the process elaborately. Okay. Okay. See, what is RNA polymerase? RNA polymerase is an enzyme which is responsible for RNA synthesis because the name itself is the RNA polymerase, the enzyme to synthesize RNA. Okay. It's a DNA dependent RNA polymerase. Why it is called as a DNA dependent RNA polymerase? Which will recognize the DNA sequence and it synthesizes the RNA strand. So that which will call it as a DNA dependent RNA polymerase. Okay. The prokaryotic RNA polymerase is a multiple subunit of 480 kilodalton. So this is the weight of our RNA polymerase. Okay. So this is the mechanism of RNA polymerase. This requires no primer for polymerization. For synthesizing RNA, there is no primers needed for polymerization. Requires DNA for activity and is most active with the double-stranded DNA as a template. So as I told you already, so the RNA polymerase which will recognize the double-stranded DNA and moreover specifically recognize the promoter sequence, okay, which will present on the DNA. So the direction of synthesis means what? RNA polymerase which will going to synthesize what? Your RNA. These are mRNA. So the synthesized RNA should be a 5' to 3' direction. Require For these synthesis, the RNA polymerase will require magnesium ions for RNA synthesis activity. Okay, lacks 3' to 5' exonuclease activity, and the error rate of nucleotides incorporation is 10 power 4 to 10 power minus 5. So what this stated means, exonuclease activity. So this RNA polymerase doesn't have the reverse. So its synthesis direction is 5' to 3'. So it lacks the 3' to 5'. Reverse exo means outside nucleus. So it has the activity for reverse mechanism. So usually are multi-subunit. So I'll explain the subunits. Okay. So see, has two alpha subunits, beta, beta dash and omega. So this is the core RNA polymerase. When the sigma factor which will come and enter into the, this is the core subunit, come and enter into the core subunit, and the RNA polymerase will call it as a holoenzyme. So as I told you already, once the phosphodiester bond synthesized, the sigma unit will automatically, which will fall down from the RNA polymerase. So what we call it as a core enzyme. So this core enzyme only will enter into our elongation process. So now you know the RNA polymerase subunits, structures, and what is core enzyme, holoenzyme. Okay. See, this is the weight of each subunit: alpha, beta, beta dash, sigma, and omega. Okay, and the functions. Each subunit has the unique functions. See, alpha subunit has which determines the DNA to be transcribed. Okay, and beta subunit catalyzes polymerization. Beta D binds and opens the DNA template, and alpha, which is recognize the promoter for synthesis initiation. So this is most important here. So when the sigma subunit which will go and bind to the core enzyme, then only the sigma subunit will recognize and bind the promoter region. After that only the what is that, the polymerase enzyme will go and do the polymerization process. Okay, omega subunit packing. So omega is responsible for subunit packing. Okay, so now you all clear. See, this is the here what I am going to show means this is the promoter region. So just imagine this is the double stranded DNA. Okay, 5' to 3', 3' to 5'. So this is the double stranded DNA. And as I mentioned you already, minus 35 upstream, minus 10 upstream, plus 1 is the downstream. So from here to there, this is our promoter, entire promoter region. Okay, and minus 35 is a TTGACA, and minus 10 is a TATA add. You simply call it TATA add. Within that only your promoter is present. So after recognizing these promoter regions, then only your transcription will start here. So this is the transcription start site. Okay. After that, the templates RNA transcript will synthesize. Okay. Now we'll move on to the elongation process. The release of alpha subunit causes the conformational change of the core enzyme. So as I mentioned you already, so now the DNA polymerase has the core enzyme, that is the conformation code for alpha subunit removing process. The core enzyme slides on a DNA template toward 3' end. So toward the 3' where the 3' end is present, so the core enzyme which will slide on to the DNA template towards the 3' end. Free NTPs are added subsequently to the 3' overage of the nascent, your newly synthesized strand. Okay, once the core enzymes will slide towards the 3' end, at there what will happen? Your NTPs, your nucleotide phosphates, will add on to the 3' hydroxyl end of your nascent RNA, that is our newly synthesized RNA. And RNA polymerase DNA segment of 40 nucleotide and the nascent RNA from complex called the transcription bubble. So RNA polymerase DNA segment. So these two hybrids form a transcription bubble. The 3' segment of nascent RNA hybridizes with the DNA template. Its 5' end extends out the transcription bubble. See, listen carefully. The 3' segment of nascent RNA hybridizes with the DNA template, and the 5' end of the nascent RNA extends out the transcription bubble as the synthesis processing. And the processing of nascent RNA, your 5' end of your nascent RNA tail is come out from the transcription bubble. Okay, see, after seeing this only you clearly know. Okay, this is the transcription bubble. Okay, non-template strand, unwinding means open, rewinding means again it will compactly closed. Okay, it's a rewinding, it's a unwinding. Okay, this is your nascent RNA. What is that? This is your RNA polymerase. Okay, and RNA DNA hybrid. Okay. So here what will happen means once the sigma factor which eliminate from the holoenzyme, core enzyme, it will move towards the 3' direction. From that DNA RNA hybrid will form. At that position it will form a transcription bubble. So here this is your newly synthesized nascent RNA. Okay, so this nascent RNA 3' to 5' direction it will synthesize. So in that condition it will form one, what is that? The DNA RNA hybrid, 8 base per DNA RNA. Okay, it will happen. So in that time it will form a transcription bubble. So your 5' end of your nascent RNA will come out from the transcription bubble. So that only they stated here. See, 5' extends out the transcription bubble as the synthesis process processing. When it's not completely synthesized, when the synthesis is completely processing, your 5' end of the nascent RNA will come out because which will already synthesize. So the synthesis process which will go on this direction, so it will continuous. Okay, so that is the elongation process. So direction of transcription, at what direction the transcription going? 5 prime to 3 prime or 3 prime to 5 prime. So this is the already synthesized RNA. So this is the complete one, and this is the going to be synthesized. So you'll tell what is the direction. Any idea? Okay, I'll move on to the next class. Next slide. RNA polymerase. C. This is the pictorial representation of RNA polymerase binding with the promoter sequence. As I told you already, minus 35 to minus 10 is your promoter region will present. So the RNA polymerase which will automatically recognize the promoter sequence, and then the DNA strands will unwind. Okay, see here, it will open up. Okay, after that only your initiation will takes place here, transcription initiation. Okay, after the initiation will happening, your sigma will go off from the polymerase enzyme. Now the enzyme is called as a core enzyme. So before we will call it as a holoenzyme. So after removal of sigma, the elongation will continuous. See, the elongation will go on this direction. The nascent RNA come out from the transcription bubble. Okay. After what will happen? So this is the hanging. The structure, it's look like a hanging structure. So what will happen? The cells nucleotides will form a hairpin loops. It will form some bond like structure. It looks like a hairpin loop. Why it is called hairpin loop? And then terminate the entire transcription mechanism. So now we'll move on to the transcription mechanism. This is our last process in transcription. The DNA polymerase stops moving on the DNA template. The RNA transcript falls off from the transcription complex. The termination occurs in their row dependent and row independent. Nothing. They simply tell the RNA once the termination will start, the RNA polymerase stop moving the DNA template. Okay. RNA polymerase is responsible for synthesizing or RNA. Okay. So once the termination will start, what is the role of RNA polymerase? So automatically it will stop their moving on DNA template. So that's why they are stating here. So the RNA transcript fall off. RNA transcript means what? Transcript fall off from the transcription complex. So what is that here? RNA transcript is our newly synthesized mRNA that will automatically fall off from the transcription complex. For this process, we need two proteins, that is row dependent and row independent. Okay. Termination function of row factor. Row dependent termination. So as I told you already, once the transcription bubble is here, the newly synthesized RNA will hanging like this at 5 prime end, and the synthesis go on this 3 prime direction. Once the elongation is completely synthesized the mRNA, it will move on to the termination process. So what will happen means a row protein will go and bind the newly synthesized mRNA. Okay. The row factor, a hexamer. Hexamer means it has 6 atoms. is a ATPase and helicase. Is a ATPase or and a helicase. It is composed of ATPase and helicase enzymes. And the row independent termination. The termination signals is a stretch of 30 to 40 nucleotides. See, here what will happen, the row protein which carries the 6 atoms of that is a hexamer of ATPase and helicase molecules, which will go and bind to the hanging strand of your newly synthesized mRNA. So what will happen, the row protein already having 6 molecules, so it will automatically go and bind. The weight will pull out the newly synthesized RNA. So this is the row dependent mechanism. And here this is the row independent termination. The termination signal is a stretch of 30 to 40 nucleotides on the RNA transcript consisting of many GC followed by a series of U. See here, it's a stretch of 40 to 30 nucleotides on a RNA transcript. From 40 to 30 nucleotides on RNA transcript is nothing but your newly synthesized mRNA, which consists of many GC. So as I told you already, the newly synthesized RNA has a more GTP than the ATP in the previous slide. If you remember that, it will help here. So consider many GC followed by a series of U. So at that stage, the sequence specificity of this nascent RNA transcript will form a particular stem loop structure. So carefully listen. At this sequence, many GC followed by many U's. Okay, many U's followed, GC will followed by many U's. After many U's, the GC will appear. At that sequence stretch, your nascent RNA transcript will form a particular stem loop structure, okay, to terminate the transcription. See, this is our hairpin. Look, which will show you. I will previous slide. I will show. I show this image in previous slide. No, where it is. See, this is. So how this form means is a row independent mechanism. So, see, this is how elaborately they are showing. See, here this is your synthesizing. See, the 3' end and this is the 5' end. This 5' end will come out from the transcription factor, and the 3' end is inside your template, which will bind with your template. So see, this is the many U's. The GC is okay. So GC is followed by many U's. When the sequence will come, the transcription elongation will automatically stops. So at this stage only, the loop will form. Okay, the loop will form at this stage. Okay, so see here. See, this is a stretching sequential stretching. So when you just fold it like this, the C is here, you just fold it like the sequence will like a stem, and this will like your loop. See, this and this like your stem, and this sequence will form a loop. Your first and last sequence is your base. You just imagine when you're stretching one thread and you form you just hold the two ends and you make this kind of loop. What will happen? This actually happens here. So at that position only it will form a hairpin loop structure. So transcript folded to form termination hairpin. So what will happen? Automatically it will terminate the transcription. So this is the entire process. Okay. Now the transcription of prokaryotes will come to the end. So these are all the inhibitors which is used for transcription. Rifampicin, streptolidin, actinomycin, cordycepin. These are the some antibiotics which will inhibit transcription. Okay. So I'll roughly explain again. This is your promoter region. Your DNA polymerase will recognize the promoter region. Sigma factor which is present on your DNA polymerase. Sigma factor is a subunit which has the specific capacity to recognize your promoter region. After recognition, your double strand will unwind and the transcription initiation takes place. After that, your sigma will goes off, your elongation starts. So now we call it as a core enzyme. So after the elongation will go on, and then your what has happened? Your termination will happen. We had two kind of terminations, or one is row dependent and another one is row independent. Okay. So girls, thank you for your kind cooperation. So I'll send you the slide and notes. Be prepared for your exam, and thank you so much for your patience, girls. Thank you.